Well, I first would like to have a very short summary of the sessions of yesterday and the most important conclusions that I think we could make. That is, first of all, with respect to the GMP inspection, there are quite some changes over the last years, and this is also reflected in the drug regulatory agencies here in Saudi Arabia on the one hand and in Jordan on the other hand, where really high standards are being set with a focus obviously on the quality, the safety, the efficacy, availability and affordability of drugs. And I think what I have gathered from these different lectures is that there is also a strong need for a unification of the regulatory agency within the entire MENA region. If we then look at the quality management within the drug sector, then it's clear that we are moving towards a life cycle management of drugs, and also there seems to be quite a need for a worldwide harmonization. It was also clear that we should try to go to a lean management, but still taking care of the fact that quality of a drug is an absolute property. I mean, there are no compromises to be made. We were also made aware of the fact that naming and labeling is a very important issue, and that can be simply overcome by taking care of some particular guidelines that we should take into account to avoid errors in use of medications. And it's also such that, again, this is actually a global problem that should be attacked globally. And last but not least, it is also clear from the last sessions yesterday that health technology assessment, pharmacoeconomic analysis, that this all should also be included in the general health policy. So far the short summary and conclusions of the sessions of yesterday, and now I would like to go with you over some general key concepts with respect to biologicals and subsequently biosimilars. And just to remind you that if we talk about biologicals and biosimilars, the point is that we are actually depending on the use of living systems to produce those drugs. So we will also see that the properties and the characteristics of these biological medicinal products will depend on particular properties of these living systems. And the first thing I would like to draw your attention to, that if we talk about biologicals, usually in the pharmaceutical sector, these will be recombinant proteins that have been produced. And to produce these in a consistent manner, you actually, as a company, you have to generate a cell line that includes, and this is indicated in green here on these circles, that includes the genetic information that will guide the cell to produce the protein. The production of a recombinant protein for pharmaceutical use, for drug use, is actually a pretty complex process. It's not just simply the cell. The cell has to be stored in a stable way. It has to be able to recover from the liquid nitrogen, to be cultured in big volumes, up to 10,000, 20, even 40 or 50,000 liter bioreactor tanks. And since these cells are living systems, you will appreciate that it's very important to take care of these living creatures in order to make sure that they are constantly in a good, optimal environment for their own sake and also for the sake of an adequate protein production, the protein that we want the cell to produce. These bioreactors is one step in the entire chain. Obviously, we have to purify the protein, we have to characterize it, and so on. If we talk about biologicals in the drug field, it's also important to realize that we have quite some differences, biologicals versus typical chemical drugs. And in this typical slide that you may have seen before or something similar, we have here on the left-hand side a very small dot that you may be able to see. That's acetylsalicylic acid, the active compound of an aspirin, for instance, which is a very small molecule and a very simple molecule. Then in the middle we have interferon, which is a biological molecule, which you can see, which is much larger in size, a bit more complex. And then on the right hand side you see the size, the relative size of a monoclonal antibody, which is approximately 1,000-fold larger in size and much more fold with respect to complexity relative to a typical chemical molecule. We will also have to take this complexity into account when evaluating the properties and the physicochemical characteristics of such a molecule. It's not only the size, it's also the way they are produced. Chemical synthesis for a typical chemical, low molecular weight molecule, and then obviously cell cultures. And this also has a consequence to the composition of your final material. You know, when we talk about acetylsalicylic acid or paracetamol, we will never discuss the structure. There's only one molecule that is acetylsalicylic acid. No variations on this molecule. Whereas for a protein, we should realize that they are composed of a heterogeneous mixture of different variants of the same molecule, and that the relative appearance of a number of variants is very much dependent on the process that has been used. And just to give you a very brief example of processes that do have an impact on this heterogeneity, and a typical one is glycosylation. You may recall from your courses biochemistry that a protein, when it's being produced during a certain step in the cellular production process, sugar moieties or sugar-like moieties are being attached to particular amino acids on this amino acid chain that establishes the protein. This glycosylation is important for the stability of proteins, for the correct folding, and in many cases, especially for monoclonal antibodies, glycosylation is also important for the functional properties in vitro, but also in vivo, and thus may have an impact on the clinical efficacy of your molecule. Glycosylation is one cause of heterogeneity within these proteins, but there are also a number, as you can see here listed, a number of other enzymatically controlled processes that do occur within the cell during protein synthesis, and that lead to this heterogeneity. And this is dependent on the cell type, a bacterial cell, a yeast cell, a mammalian cell. They will all behave differently with respect to these different enzymatic reactions. And it's obviously also dependent on the culture conditions, because enzymatic reactions depend, for instance, on the temperature, on the pH, on the presence of particular catalyzing metal ions, and so on. It's not finished once the protein has been released by the cell. There are also chemical modifications that can occur at certain amino acids, such as, for instance, a typical one, the third bullet on the right hand side, oxidation. Methionine is an amino acid that is very sensitive to oxidation. You always have some oxygen, not only in the air, but also in your buffers that you are using. So don't be surprised if you characterize your protein. There will always be a certain degree, a certain number of methionines that are oxidized to a certain extent. And again, this adds to the heterogeneity of your single molecule. And this is dependent, obviously, on external factors. And if you calculate from a theoretical point of view, you may end up with approximately 100 million variants of the same molecule, which you will probably understand makes the analytical evaluation of your protein a little bit more complex than that for a typical chemical molecule. In addition, when we administer those proteins, those biological drugs to human patients, we should realize that any biological, even if it's a human recombinant protein, it may elicit an immune response in the patient. So that's also something we have to take into account. So far the intermediate conclusion is that product variants in a protein population will always be present, that from a theoretical point of view we have a large number of possible variants, 100 million. We will not be able to recognize all those different possibilities, but if there is a theoretical amount of 100 million, you will not be surprised if you can detect easily 20 to 30 of these variants from an analytical point of view. So the only way to guarantee reproducibility in your production process is not only to look at the analytical data at the very end, but also to make sure that the entire production process can be repeated in a consistent way. And that's why you will often find a paradigm that when we talk about a biological medicinal product, it's the entire process that will determine the ultimate properties and characteristics of the product. So we need to know the entire process. What does this mean in real life when we are dealing with drugs? And here I took the example of infliximab, for instance. You will see here on the right-hand side on the slide, the top line, for instance, we have the cDNA in green that encodes for the protein, the antibody infliximab, which represents here in this first line the originator molecule, for instance. And then if the patent has expired, other companies may decide to also produce infliximab. The only thing they know from the patent is actually the DNA sequence that can be used. So in these different processes, these three different cell lines that will be made independently, the only common thing is the green symbol that represents this cDNA. So the genetic material of the encoded protein will be exactly the same, but the cell will be different. The process, the arrows that you can see here, the process, the purification steps will be completely different. But since you use the same DNA sequence, you will end up with the same protein, infliximab, in all three cases. And when you look at the very first glance, it all looks like infliximab. But if you look into a little bit more detail, you may notice that infliximab is not written in exactly the same way. So they look the same, but they are clearly not identical. And if we are lucky, they may be sufficiently similar to each other. This brings me to the word similar and biosimilar. We will call those copies of biological molecules. Once the patent has expired and another company also makes the protein, since they are not identical to each other, we will call them similars to the reference product or biosimilar. And here we have a definition of the European Medicines Agency, where you can read that a biosimilar is obviously a biological product that actually contains a version of an active substance of a product that has already been authorized in the European Economic Area, and for which it has been shown through a very extensive comparability exercise that this version is similar to a reference product. And as you know, within the European Medicines Agency and European Area, we have experience with biosimilars since 2006, when the first guidelines had been established. Obviously, also other regions, WHO, established some guidelines, and in the meantime, all the most important regions within the world established their guidelines with respect to the registration of biosimilars. And you will also see here that Jordan, the Saudi and Arabia FDA also have their guidelines. I won't go into too much detail for those guidelines, but it's clear in the world of drugs, it's all about quality, safety and efficacy of these molecules. And this is also linked, since these are copy versions, we need a extensive comparability exercise, and if you want to compare something, you obviously need a reference product, and it's important that the reference product needs to be an authorized reference product. So how do we start with the development of a biosimilar? The first thing is that you need to establish your process. You will establish your cell line, you will set up a purification process, and then you will compare your molecule with the characteristics of the reference molecule. You will do this at a physicochemical level, structural properties, biochemical properties. You will also do this in vitro with respect to its functional biological properties. And in the first round, you may be sure that since you have not sufficient information on the process that the originator company is using, you will probably find differences between your product and the reference product. This is not a disaster. You will just have to look what are the differences, then in a rational scientific way think how should I modify my process to make those differences smaller. You will go back to the bench, adapt your process, start with the characterization again, do the comparison, and you will hopefully notice that the differences become smaller. Maybe not small enough to be acceptable, you may have to go back again. So it's a reiterative process in order to get finally to a product that is highly similar to the reference product with respect to its analytical properties. And once that is the case, since you have demonstrated from a structural point of view and an in vitro biological point of view, then you will be allowed to introduce less non-clinical and less clinical information for your registration file. Why? Because we expect, since the molecule is very similar at the structural level, we may expect to a certain degree that the clinical and non-clinical properties of this copy of this biosimilar will most likely also be very similar to that of the reference process. So the clinical evaluation, which will be discussed in the next lecture by Professor Till, will only serve not to prove the efficacy, but merely to confirm the biosimilarity at the clinical level. If as a company you want to start producing something, it's good to have some guidelines to know what the regulatory authorities expect from you. So, and here I have an overview of the different guidelines that are available within the European Medicines Agency. And what may be the most important thing here is that at the bottom you will see that apart from general overarching guidelines, there are also some product-specific guidelines. Because you will appreciate that we will not look at the monoclonal antibody in the same way as we look at the simple molecules such as insulin, for instance. Now, what is actually required if we compare the registration requirement for an original molecule? We have obviously the three main components of a registration file: the quality dossier, the CMC, the non-clinical part, and the clinical part, where we have all those different sections on the one hand. If we now look what is required for the registration of a biosimilar, if you look at the left-hand side, you see that everything remains in dark blue, so this needs to be submitted. And in addition, you have indicated here in red the extensive comparability data at the structural level. So it's important to realize that there are no shortcuts with respect to the quality that needs to be demonstrated. That's, I think, obvious in a drug sector. As I mentioned before, in the development, if you have shown from a quality point of view that your product is very similar, then you will be allowed to restrict your non-clinical and your clinical part. The most important thing here, and we will discuss this later in the session, the most important thing here with respect to the clinical point is that you may notice that you only need studies in one indication, and then we will see later on how we will deal with the approval for the other indications based on the concept of extrapolation. Since we have less clinical information at the time of approval of the product, it will also be important to demonstrate post-marketing the safety in larger population, possibly to confirm the efficacy in the other indications, and obviously also follow up the potential immunogenicity of your product at a longer time in larger study populations. Where are we currently in Europe? A number of molecules have been refused for a number of reasons. I won't go into detail. It just illustrates that the process is pretty rigorous. It's not just making a copy and submitting your file. You will really have to show the data that everything is okay. There are approximately 41 dossiers that have been approved by the European Medicines Agency. And there are still currently 13 under review, and since for a number of other molecules patents will also further expire, this will constantly be going on with respect to the submission of new biosimilar files, I guess. What is actually needed? Just a snapshot of, for instance, a number of analytical methods to which the first infliximab biosimilars had to be subjected to get approved, and I won't go into detail, but you will see here on the left-hand side, for instance, N-terminal sequencing, C-terminal sequencing, circular dichroism to confirm and to compare the secondary structure, and so on. On the right-hand side, somewhere in the middle, you will see glycosylation as one of the important parameters to evaluate, especially for a monoclonal antibody. Glycosylation is also a very complex process, and you may also notice that it's not just glycosylation. You have quite a number of different sugar-like moieties that can be attached, and that will have to be evaluated in much more detail. Now, the structure is one thing. You will also have to demonstrate, for instance, in the case of a monoclonal antibody, that actually your antibody binds to the target where it's supposed to bind. TNF infliximab, for instance, you will have to demonstrate that it binds to TNF-alpha. What is important, especially for monoclonal antibodies, that these molecules do not have one single domain that is important for its function, but also the constant region, the Fc region, that we see here on the molecule. The Fc region is also very important and may have also an impact on the clinical properties of the molecule. So you will also have to demonstrate that your biosimilar infliximab binds to the different Fc receptors in the same way, to the same extent, with the same affinity as the reference infliximab molecule. And as you may know, it's not sufficient to bind to the target, to bind to the Fc receptor, but you also expect, subsequent to this binding, the same functional effects. For instance, the neutralization of TNF-alpha when your biosimilar infliximab binds to TNF-alpha. And it's not sufficient to demonstrate that your antibody binds to Fc receptor. You also have to demonstrate that subsequent to the binding to these cellular receptors, you induce signaling pathways within the cell that then may lead to those functional properties such as antibody-dependent cellular cytotoxicity, complement activation, and so on. So these are different things that you will have to consider and make life as a biosimilar developer pretty tough. I mentioned also at the beginning that we will never be able to make an exact copy because of this heterogeneity, because of the fact that we depend on these living systems. If you look at the European Public Assessment Report, so the things that you see here on these slides is all publicly available information that you can extract from the European Public Assessment Report, you will notice, and a few words are indicated in red, where they find some differences between the biosimilar and the reference product. So, from an analytical point of view, don't be depressed if you find differences. The important point and the challenge is to be able to demonstrate to the regulatory authorities and to yourself that from a scientific point of view, or you have scientific data or scientific background information, that these differences that you may observe may not have an impact on the clinical properties of the molecule. So that's very important to realize. Now, two examples from data, again from the European Public Assessment Reports, to demonstrate that the decision on the statement whether, or on the conclusion that the biosimilar that is being submitted is indeed sufficiently similar to the reference product, this is not always a black and white situation. If we look here, for instance, for a biosimilar infliximab, SB2, you will find in the dossier, typical for infliximab, you should know that this is a chimeric molecule, and typically approximately 40% of the patients do develop antibodies against the drug within the period of the first 3 months, for instance. So this is very typical. We know this for the reference product, so it's obvious that we also expect antibody-drug antibodies, anti-drug antibodies also for the biosimilar. What they did find with the biosimilars that 47% versus 38% for the reference product developed antibodies. And obviously, antibodies against your drug may have an impact in quite a number of cases on the efficacy. And there was some discussion within the scientific experts of the committee that had to give an advice on this biosimilar. And actually, from 14 out of the 36 experts decided that actually the potential impact of the antibody formation against the drug infliximab, that actually the impact on the efficacy was not unambiguously clear. So they actually were in favor of not approving this molecule as a biosimilar. But, you know, in a democracy, it's a majority that decides. Since the majority was in favor, it was actually approved. I'm not saying that this is a bad biosimilar, I'm just saying that there may be some residual uncertainty that may lead to discussions. In addition, one should also realize that these data were obtained in a patient population that also received methotrexate. And methotrexate is an immunosuppressant. So you may even, in this case, the difference may even have been underestimated because you were looking under conditions where you had an immunosuppression. Another, and especially as a scientist, I like this one. This is for an etanercept biosimilar, where they... My battery is running low. Okay, 2 minutes, yeah. You will notice here, and I won't go into too much detail, but the first statistical analysis resulted in a much lower immunogenicity or apparent immunogenicity for the biosimilar relative to the reference product, 1% versus 13%. And then they ultimately decided, yeah, this is a huge difference. So the biosimilar is actually not very similar to the reference product. And then they decided in the analysis that for some reason that they should or might discard selective data obtained from week 4 and week 8, and they did a reanalysis, and then they found a difference that was much smaller, 0.3 for the biosimilar, 0.7 for the reference product. But it's obvious that as a scientist, I would never get away with this selective omission of data to get my paper published, obviously. So this is a scientific issue. Very quickly about the terminology. And why I'm saying this terminology, you know, we have the reference product, which is here on top, and then we have the biosimilar, for which we have some differences. I won't discuss the second generation or next generation molecules, but you may notice here at the bottom, we also have, within different regions in the world, what we call non-comparable biologicals. These are just molecules that have been made, and for which we have no idea on the quality. I'm again not saying that this quality is per definition not okay, but these molecules have never been compared with an authorized reference product. And here I have a typical example. I won't go into the technical details. This is a real biosimilar. Within the European regions, this is the reference product profile, and this is the biosimilar profile. You see that the profiles are pretty identical. For a similar molecule, this is again the reference product, and here these are different molecules of erythropoietin that have not been compared to the reference product, obviously, but that are on the market somewhere in Thailand here, if I remember correctly. And you see that these are definitely not adhering to the high standards that we put to biosimilars. Just a few examples. Reditux, India, a difficult name, Yi Saipu in China, for instance. These molecules are not to be considered as possible biosimilars. So, to conclude, if we look to biologicals in general, these are very complex and often multi-domain molecules. They are characterized by an intrinsic heterogeneity, for which these properties are dependent on the entire process. We will discuss interchangeability later. And if we look in particular to biosimilars, it's clear that the concept is very well defined, and that the process for approval is a very rigorous process if we are talking about a true biosimilar. An important point that I would like to stress: there is no doubt about the intrinsic pharmaceutical quality of a biosimilar. But we should realize that there will always be some differences between the biosimilar and the reference product, and that the major challenge in the approval process is to identify whether or not particular differences may have a clinical impact. So we should realize that when we implement the use of biosimilars, there will always be some residual uncertainties that we will have to deal with and live with. So that's why also an adequate pharmacovigilance is also, once a biosimilar is being introduced onto the market,